My name's Eddie Rubin. I'm the director of the JGI Institute, and it's really a pleasure to welcome you to Genomics of Energy and the Environment user meeting. This is this is our 10th anniversary for having this meeting. Each year the meeting has grown. Usually we get most of our registrants in the last 2 weeks. 2 weeks ago we got a call from the fire marshal from the hotel saying that they ran out of space. We had to shut off registration. And so though there may be a seat next to you, it probably won't be available tomorrow. And so next year we might be doing this at a different place to accommodate the growing interest in genomics and how it applies to various energy and loosely related to energy and environmental issues. The meeting's purpose are several-fold. One of the most important ones, obviously, is to hear great science and to see science both in the talks and the posters. Also, the JGI is first and foremost a user facility. And so this meeting serves as an opportunity for many of the users who make up the biggest percentage of this audience to interact with the scientists at the JGI that are doing your projects. And so this is your chance to chat with them. Also, the JGI continually is evolving, and many of our directions come from the input that we get from users. And so I urge you to grab me and grab other people who have JGI sticker and tell us where you think the JGI should go, what sorts of capabilities that would enable your science in the future. And also, you'll get to hear about some of the new capabilities that we're developing. You can see how they fit with things that you would like to do. Okay, so a few announcements. The exits are in the back. They're guys with red jackets. California shakes sometime and things come down, and so that's where you go. And there are people with red jackets that'll usher you out. There is poster sessions, both tonight and tomorrow night, and there is food associated with the poster session. There'll be, there's a bar tonight. There won't be a bar tomorrow night. Tonight's will be upstairs here. Tomorrow there will be a poster session at the JGI, and there'll be buses that'll take you, it's about a 10-minute trip from here to the JGI, and there'll also be food there. And so tonight's session will conclude with, we have 3 plenary talks, short talks, but it will conclude with an award. The Van Niel Award will be presented here by a visitor from University of Queensland to a participant, and you'll get to hear that at the end of the session. And so the science tonight, the Department of Energy funds 3 bioenergy research centers, and those centers are focused on the conversion, the basic science, the conversion from sunlight to plants to sugars, eventually to biofuels. And there's one in Berkeley, there's one based in Wisconsin, and there's one based in Oak Ridge. And we're fortunate to have representatives from each of these centers to tell us about different things, exciting science going on there. And so it's a pleasure to have as the first speaker Blake Simmons, and Blake is the Chief Science and Technology Officer and Vice President of Deconstruction in the Berkeley Center, which is called JBEI. Blake has a passion for breaking down plant material into sugars. And he'll talk about that on function-based genomic screening for the discovery and manipulation of lignocellulosic enzymes and pathways. Okay, great.
So thank you, Eddie, for that introduction. Can everybody hear me? I have a tendency to speak loud and fast, so hopefully I won't blow anybody's ears out with this. So my name is Blake Simmons. I'm your JBEI ambassador. That's the Joint BioEnergy Institute that's located in Emeryville. It's a partnership of 7 different entities whose primary mission is to drive the science around how do we understand better ways to break down lignocellulosic biomass and convert it into renewable fuels and chemicals. And as Eddie stated, I have a passion around biomass. Particularly, I hate it. I grew up in Nebraska. My whole family are farmers. I don't like corn. I don't like irrigation. I don't like raising things that are green. I have artificial grass at my house. And so I really like to break down biomass. I'm really good at it. And my team is exceptionally good at it. And I'm going to be walking you through the stages of what we've been doing around ionic liquids primarily. But Eddie also asked me to talk about biofuels and what are they good for in today's $50 a barrel oil regime. So, I don't think I need to preach to the choir here, but biofuels are important for several reasons. Not the least of which is the specter of climate change. And we're already past the point of seeing climate change. And this is just the compilation from NASA of the global temperature mean over the past 60 years condensed into a short movie. So we're already seeing a warming on the planet, and it's primarily associated with greenhouse gas emissions. And there's a great paper that came out just recently from Lawrence Berkeley National Lab where they actually did the experiment where they proved the equivalent that gravity exists. And that is that CO2 in the atmosphere causes a change in the climate, and particularly atmospheric warming. And biofuels can help address that by reducing closing the balance on the carbon cycle, in that the carbon we emit from the transportation sector, which is 97% derived from fossil fuels, is now the carbon source for photosynthesis that we can take and put into plants, liberate the sugars from it, convert those into fuels, and then put them in the tank. And so it's all about closing that carbon balance and trying to reduce the overall carbon spread from it. Now, we have some emission drivers here, such as the Renewable Fuels Standard, which is an offshoot, well, a direct implication of the Energy Independence and Security Act of 2007, that set forth some very ambitious goals for the production of advanced biofuels with a 15 billion gallon a year cap on corn ethanol. Not all biofuels are created equal. Okay, some are better than others, and ethanol is a great gateway fuel to the future in that it can get us over the hump and get into advanced biofuels that are drop-in replacements for gasoline, diesel, and aviation fuels. And so that's that 21 billion gallons that you see, the delta here, or over here, that is beyond the corn ethanol mandate. The problem is that the advanced biofuels community suffered through what we can quaintly call the Second Great Depression. The capital market dried up. There was very little investment in the big steel projects for a period of about 5 years. And so if you take a look at the biofuel mandate that was part of RFS2 and the current capacity, it's lagging behind. And that's good news and bad news. The good news is that there's an opportunity and a driver here for fundamental advances in science and technology to enable the commercialization of advanced biofuels and get us to those targets even faster. And so that's even a better motivation for the BioEnergy Research Centers to exist and to help drive the future towards this advanced biofuel outcome. In addition to fuels, we're not only talking about getting off petroleum for the transportation sector and the 240 billion gallons a year that we consume in the United States, but it's also about how do we displace the whole barrel of oil. And how do we do things in terms of renewable chemicals that can also offset why we need to pull things from the ground in the form of oil and crack it into everything that we use. And what we derive from oil and petroleum is basically everything. It touches every aspect of our life today. And so it's a very Herculean lift to replace the petroleum that we derive all of our benefits from today with a renewable resource. But again, that's mission central to the BioEnergy Research Centers. Now, we've experienced over the past 6 months or 8 months a kind of a wake-up call for the cellulosic biofuels community, in that the price of oil on a per barrel basis has dropped pretty significantly. It's between $50 and $60 a barrel now. I think it might be $63.75 today. And so, this is almost an exact repeat of what happened in Biofuels 1.0 with the original oil crisis, or the first oil crisis we termed as such, at the late 70s and early 80s, where suddenly OPEC found a sea of oil and they turned on the taps and the price dropped and biofuels were wiped out, right? So do we do the same thing again? We just say, okay, well, climate change is important. Yeah, we don't want to destroy the planet. We want to save it. Well, but we really like cheap oil, so we'll just put our head in the sand and hope for something better next time around. Well, I'm a big champion of not doing that, and I think there's a lot of people in the audience that share that same passion. And if you really take a look at the true cost of gasoline, it's not only the production and conversion costs, it's also the externalities associated with climate change and others that can, create a price equivalency, if you will, that ranges anywhere between $5 and $15 a gallon. And that is the true cost of carbon. And there's a lot of hotly contested issues around that, but it creates still a mission driver and an impetus for the advanced biofuels and advanced renewable chemicals to come from lignocellulosic biomass. And so, it only takes $3 a barrel, actually, to pull a barrel of oil from Saudi Arabia. There is very little else simpler in the planet than sticking a steel pipe in the ground and letting pressure push it up, collect it in a barrel, and crack it, right? That is trivial. So it's not really about meeting the production costs. It's how do you meet the systemic environmental costs and the true cost of carbon that we need to concern ourselves with. And so how do we weather this current storm? Well, again, we're going to continue to do world-class basic science. We're going to talk to industry early and often about meeting their ever-changing market needs and really focusing on co-products as well as biofuels to augment the business model and the drivers for biofuels. So we still matter. The BioEnergy Research Centers are still needed in the long term, short term, and near term. So now we get to the why I'm here today, to talk to you about the Joint BioEnergy Institute and our research program. So I have a schematic here of feedstocks to fuels, and all three BioEnergy research centers follow this kind of flow chart. You have to understand plants, what makes them recalcitrant. Corn ethanol is wonderful, and we've been doing it for thousands of years, because if you stick corn kernels in water, heat it up, and put yeast in there, you get ethanol, and we like that. Right? We've been deriving pleasure from that for a very long time, and the first Model T Ford was actually designed to run on it. But if you're going to understand lignocellulose, there's a lot of fundamental science gaps in what makes a plant cell wall recalcitrant, how do you develop engineering motifs to overcome that, and how do we dedicate energy crops that are designed or evolved or engineered for energy production. We then need to have better processes by which we take the lignocellulosic biomass and break it down into its intermediates. That's both fermentable sugars in the form of polysaccharides that can hydrolyze into the fermentable sugars, as well as doing something with the lignin. And I think all three BioEnergy research centers have a growing awareness of the importance of lignin, and that it represents anywhere between 20 and 30% of the lignocellulosic biomass, and you don't just want to throw that away. And then finally, at JBEI, we're looking at how do we go from sugars to fuels using synthetic biology, and it's really a focus on drop-in fuels for gasoline, diesel, and aviation fuels. We don't have a research program on ethanol production at JBEI. And so our approach to this is that we have a feedstocks team that's looking at the plant issue. We have a deconstruction team, which is basically sugarland. And then we have fuel synthesis, which is how you go from sugars to fuels. And then we have an integrated technologies division that provides a lot of the enabling tools, analytics, bioinformatics, computational modeling, et cetera. So we've really carved out this space around ionic liquids as our deconstruction element. And I'm starting here because this will put in context the rest of the talk and why we need to come up with better systems downstream from this process. Ionic liquids are an offshoot of the green chemistry movement. They were first touted as a replacement to organic solvents. They have very low vapor pressure. They can be in a variety of formats at room temperature, including solids and liquids. And they are basically molten salts. Sodium chloride is a liquid at 700 degrees C, but you wouldn't necessarily want to use that as a pretreatment medium. So we focus on a combination of cation and anion that is a room temperature ionic liquid that back in 2002 was reported by the Robin Rogers group as being capable of dissolving microcrystalline cellulose and generating an amorphous product. Why that's important is because it makes the enzymes that much easier, it makes their job easier to liberate high yields of fermentable sugars from cellulose and the other components in polysaccharides. And if you use the right combination of ionic liquids, you can have a very dramatic effect on the plant cell walls. And so here is a movie taken of switchgrass, and you can see it goes from coherent to incoherent. Great, well, grass is not that hard to overcome, right? They're pretty easy. What about something like eucalyptus? Well, again, same impact, same result, basically molten biomass, right? And at the end of the day, we create a process where we get very high sugar yields out. But there's a caveat to it. The way we achieve these great sugar yields is you do the pretreatment, and then you recover the biomass by adding an anti-solvent, and then you wash residual ionic liquids away, right? Because a lot of the ionic liquids that are known to be very effective biomass pretreatment solvents are not that conducive to maintaining high enzyme performance, nor are they conducive to maintaining high microbial fermentation performance. And so we only get these when we wash the buggers out of the substrate and then add the enzymes to it. And that creates a lot of challenges because you don't want to waste a lot of water. It drives up costs. And you want to be able to maintain the ionic liquid. And actually, if you could come up with a process that is a one-pot pretreatment, saccharification, and fermentation process, that would be ideal. It would simplify the unit operations and drive the costs down. But we can't use the enzymes and microbes that we have today in order to achieve that. So what we set out to do is, develop this process flow that I have here top to bottom, where you start with feedstocks, you then do pretreatment, you would then do some recovery operation, then you do saccharification, and then you would hand off the hydrolysates to fuel synthesis. But the problem is that this step here requires a lot of cleanup. So the solution here, or one potential solution, is how do we come up with a cocktail of enzymes that is tolerant to the ionic liquid and also high temperature? Because then you can decrease the amount of human intervention that you need between these two steps. And so we embarked on and this is, we identified this very early on at JBEI. And so we embarked upon a very aggressive research program that utilized not only the resources that we have within JBEI, but also the Joint Genome Institute and the Environmental Molecular Sciences Laboratory, where we went out into the real world. And we looked at a bunch of different ecosystems. And one of these is a thermophilic compost pile, where we took the initial ecosystem, and we weren't really interested in what was there initially, But we wanted to hit it with a perturbation, such as the presence of the ionic liquid or a high temperature, and we cause a dramatic reduction in the microbial complexity in the community. And then we get to the point where we can identify through metagenomics and metaproteomics what's giving rise to the function, because we found things that could tolerate the ionic liquid and still handle biomass. And so then once we've identified them, then we put them through our recombinant protein expression pipeline. In certain cases, we scale them up at the Advanced Biofuels Process Demonstration Unit, LBNL. And what we're able to get is a Jtherm, we called it, JBEI. You might notice that if you get a lot of acronyms coming from JBEI, they more often than not start with J. And so this is a Jtherm enzymatic cocktail where we can consolidate pretreatment and saccharification and still maintain very high sugar yields. And this is a comparison of untreated, the conventional process, which is the wash process with CTEC2, which is a commercial enzyme cocktail from Novozymes. We do the one pot. We've introduced the commercial enzymes into that one pot mix without cleaning the substrate. We lose activity dramatically. And when we put in the Jtherm, we actually achieve pretty much the same yields that we get, 90 to 95%, on the C6 spread, and then also on the xylyl oligomers that we get from hemicelluloses. So this is a very effective cocktail in terms of sugar yields that we get out of it, but it also has a very dramatic impact on the sustainability of the biorefinery in that we drive down the water consumption dramatically. So that means you don't have to have such a massive wastewater treatment plant, which a lot of cellulosic biorefiners are spending 50% of their operating costs in maintaining. We've also come up with a way that we can screen through a lot of different enzymes. One of this is the high-throughput NIMES technology that Trent Northern and others have done. This is basically combining surface acoustic printing. This is a movie taken of little droplets that are being spit out using that device onto a multi-plate. We can get large-scale data visualization as heat maps, and what's great is that we can tailor the substrates that we put down into this mix, as well as the enzymes that we can put down in this mix, and we can get a very easy before and after snapshot of activity. And so this is a great way where you can get automation in a very significant fashion. In fact, it's about a 10-fold increase in throughput compared to robotics. And one of our first pioneering projects, and this is a really busy slide, I apologize for it, but time is short. We actually used and leveraged the JGI Gene Synthesis capability, where we did a phylogenetic scan of GH1s, which are beta-glucosidases, the cellobiose to glucose reaction. And we picked 200 of those genes that represent a wide swath of the phylogenetic diversity. And then we got about 180 of those, 175 that were done. And then we put them into the high-throughput NIMS technology and screened them. And you can see it's we looked at archaea, eukaryotes, prokaryotes, and the metagenomic studies that we did. We then create this heat map for the data, and we put them in the ionic liquid in high temperature conditions and see what enzymes could perform well on a variety of different substrates, because you can put 10 different substrates into one microtiter well and get an independent activity profile using the NIMS technology. And what we're able to find is about 5 of those GH1s that we did not know beforehand were very active at the temperatures we cared about, and actually went into the cocktail augmentation of the glycoside hydrolases that we had. So it's a great way to discover a needle in a stack of needles and actually come up with a better cocktail at the end of the day. Okay, moving quickly into fermentation. So we've got the Jtherm question solved, right? We've got enzymes that can handle it. What about the microbes? A lot of the ionic liquids that we use are imidazolium-based. They are antimicrobial. Therefore, it's not a huge supposition or a leap of faith that they are not going to be that good to the microbes we care about for biofuel production. And indeed, that is correct. The typical microbe, yeast, E. coli, tolerate about 0.2% to 0.4% of the ionic liquid. So, I mentioned before we went out into the world and studied these ecosystems. Well, one of these was the Puerto Rican rainforest, where we planted bags of switchgrass and bags of lignin, not viable material. We weren't contaminating the Puerto Rican rainforest, but we wanted to monitor biomass deconstruction and turnover in that environment, and we monitored the system that broke down, and what broke down those materials. And what we found was a particular bug, Enterobacter lignolyticus, that could grow on lignin as a sole carbon source, could grow on switchgrass, and could also tolerate ionic liquids, which is surprising because there aren't a lot of ionic liquids in the rainforest floor in Puerto Rico, at least that I'm aware of. And we sequenced the genome at JGI in 2011, and this is work that was presented in PNAS by Jane Kudiakov. And what we found is, it had a whole host of responses in the presence of ionic liquid, but we couldn't elucidate one that gave rise to the phenotype we cared about. So Tom Rueg went about chopping the DNA up and creating a phosmid library and putting in E. coli. And then they basically put the ionic liquid onto the agar plates, and you screened for what could grow. And what we found is that 27 out of the 41 phosmids that grew sequence well, and were aligned with the SCF1 genome, which is a shorthand for Enterobacter lignolyticus. And without exception, all the inserts shared this 2.6 kb domain. It was just amazing. It just fell out of the sky into our labs. And what this encoded for was an MFS1 efflux pump that we found imparted ionic liquid tolerance to E. coli. And it also gave us a regulatory mechanism to control the expression of that pump. So you had the genes flanking this MFS1 pump that were only partly present on the shared sequence were this TET-R regulator upstream and this EAM-like transporter downstream. And what this, why does this thing do what it does on ionic liquids? Well, interestingly enough, it has close homology to methylbiologen resistance, which looks a lot like a cation. It looks a lot like an imidazolium cation in particular. And so when we finally went about, okay, great, we've had this discovery tool. At JBEI, we have a lot of E. coli engineering for advanced biofuel production. Could we put that pump into an engineered E. coli strain and produce an advanced biofuel? And let alone, lo and behold, it could. So this is E. coli that was engineered to produce bisabolene. You had the blue, which is with the IL pump, and the gray without the IL pump. And we were able to engineer an organism using this approach that could tolerate about 6 to 8% of an ionic liquid. What's more important is that we could actually get a dynamic regulation and control that the pumps are only turned on in the presence of the ionic liquid. So then you're not paying the metabolic efficiency all the time. So you could ostensibly have a system where it only turns the pump on when it needs to, when it sees the ionic liquid. And so in follow-up work that Andrilla's team at JBEI has done, they're actually able to prove that dynamic control in a very robust fashion. And Tom has since gone on and said, well, gosh, this is a very tight control of this expression. Can we use it for heterologous recombinant protein expression? And other teams at JBEI in feedstocks and fuel synthesis and deconstruction are actually using that regulation mechanism for protein regulation and expression. So it's a great thing that just kind of fell into our laps. So in summary, and I've cut it short because we have a lot to talk about still tonight, building an integrated conversion technology that needs a system of systems approach. You can't just find a perfect solution for your one domain, especially for a biorefinery where every unit operation is interconnected. We have a screening approach that's based on functional genomics that we find is very powerful, especially when you're looking for a needle in a stack of other needles and you don't know what the needle you want looks like. And we found this high-throughput NIMES technology as a rapid way to assess the genomic potential and proteomic potential of a system. And we developed a pipeline for the identification, isolation, and characterization of lignocellulosic enzymes. And we're expanding this now beyond just the GHs and going after lignin-degrading enzymes, because the same approach holds true. And finally, Dan Drell, I just want to point this out. Fundamental science and understanding biological phenomena matters. It really does. If we just took this phenomenon and observed it and didn't dig in deeper about why we saw Enterobacter lignolyticus is IL tolerant, we would have never tapped into this. And it's a very enabling tool. So acknowledgments here. We have a cast of hundreds. And finally, I'd be remiss if I didn't acknowledge JBEI. This is the group. Thank you for your time and appreciate the attention.
Enzymes from Halorite here because they're naturally adapting to high external salt?
Yes, so actually one of the projects that we did with Eddie's group was looking at halotolerant. It's a very halotolerant enzyme. It's actually halo-obligate. And we did some work on that, and what we found is that it's really not halotolerance that we can key off of in terms of the response to ionic liquids, but really a more systemic osmotolerance effect. And so what we're doing now is, and I didn't have time, is we're trying to come up with some engineering motifs that we understand the enzymes in the ionic liquids and what makes them operate more stable. And then, there's a lot of different things. One is thermotolerant enzymes are just more robust. And so if you take a thermotolerant enzyme and put it in the ionic liquid, you see a decrease in the T optimum about 5 to 10 degrees. There's also this net surface charge argument. There's also this effect that if you have a negative surface charge density that's very dense, it can have a shielding effect to some of the anions that we use. So you get an electrostatic repulsion between the enzyme and the anion, but it's not systemic. It's not predictable. It's very stochastic, and so it's still got a lot of noise in the system. But what we want to have is this, what we ultimately want is an ionic liquid that we don't need any special enzymes, nor do we need any special microbes that we can just use right out of the gate. And we've seen some recent progress on it, and we're terming it bionic liquids. And hopefully next year we can give you the story about that.
Maybe you answered my question at the end of yours was, enzymes are hard to scale, in billion-gallon processes. And so, will these bionic liquids be able to, take away the need for?
Well, there's a couple ways to respond to that. So the Jtherm process that we have, we actually scaled the microbial community and the recombinant protein expression up to the 300-liter size at the ABPDU, and we actually saw that as we scaled, the cocktail got stronger and more robust. So when we talk to folks like Novozymes or DSM or Genentech, they don't see the scale-up necessarily as a showstopper. But those are all in systems that are relatively complex. We have a fungal biotechnology group at JBEI, which is looking at how do we take the enzymes that work well for our process and put them into fungal hosts, understand what are the design rules for putting an enzyme into a fungal expression system so that you can enable scale-up. So that's one approach to it. And then the other approach is, well, why don't we just make the whole thing moot and come up with a bionic liquid that is very easy on commercial enzyme mixtures and commercial biofuel hosts. And the initial results that we've seen are pretty encouraging. Any other questions?
Okay, well,
Dan.
Oops, sorry, Dan Jarrell?
I have to ask, what is it in the Puerto Rican rainforest that would require Enterobacter to have an efflux pump, such as what you discovered?
That's a really great evolutionary biology question. And the methyl biologen resistance is something that you could equate to antibiotic resistance, but again, there's not a lot of antibiotics that are exposed in the Puerto Rican rainforest. And so that is a riddle that I would like to hand off to some environmental biologists and folks that have specialized in that. And we haven't really gotten into a deep comparative analysis of why those systems are there. But we're really glad that they are. But it's a good question. Yeah. Oh, is there one more, David?
I'm not so familiar with the field, but aren't microalgae, for example, much more efficient or faster to grow than crops? And then wouldn't there be different polysaccharides that you would have to tackle?
Yeah, so there is a community within the advanced biofuels focused on microalgae and actually macroalgae as well. And so if you view them as a little photosynthetic factory that you can harness effectively and solve the water problem, because the algae, even if you get a bloom of them, they are typically very dilute compared to terrestrial biomass feedstocks. And so the costs are generally higher for algal-based biofuels relative to terrestrial over the short term. There is an elegance and a robustness to the philosophy around using algae because you don't have to grow them on land that necessarily compete for feed or food, although that is somewhat of a red herring argument when you actually go to a county-by-county map resource allocation for lignocellulosic biomass feedstocks, and the BioKDF at Oak Ridge has done a great job of providing the community tools to understand that. But algal biofuels are worth pursuing. It's just going to be a longer stretch because the genetic transformation tools, the process engineering, all of that is not relative to the state of the art in lignocellulosic biofuels. And just point in fact, a lot of the macroalgae and microalgae community, the industry players, are selling products that they sell for $1,000 a kilogram, and some of them are going under. And so if they can't make money at $1,000 a kilogram for their product, selling something for $3 a gallon, or $3 a kilogram, right? You're several orders of magnitude away from viability. So the challenges are significant, but they are worth going after. And so there is still a rather robust research program in the broader community and within the national labs, academia, and industry that is trying to address just that.
Great. Well, thank you. Another one more?
We need a...
Okay.
Just going back to the previous question about digging into environmental science, so why did you choose to go look in the Puerto Rican rainforest in the first place, and did you look anywhere else?
One man, Terry Hazen. Okay. He has a connection with, he was the director of microbial communities before us. He had a very strong collaboration with the Long-Term Ecological Research Institute that was NSF funded in Puerto Rico that we leveraged and took advantage of, and it was just an easy site for surveying. The treaties were all in place. There was no question about could we use the genes that we discovered there or not. And so it was a target of convenience, if you will, but also scientifically compelling because it has some of the highest turnover in biomass rates reported on the planet. That's not to say that there aren't environments that have higher rates, they just aren't reported yet. So it was a very compelling site for us for several reasons, but primarily it was because the passion and scientific caliber of the man who was driving it, and that was Terry Hazen. So full credit to him. Okay.
Okay, thank you very much, Blake.